Hello children. As we are discussing the chapter 2 of your book, Microorganisms, Friends and Foe. In our previous 2 videos, we have discussed certain topics. We'll revise them again. The first is, what are microorganisms? As the name of the chapter indicates, what are microorganisms or microbes? The living organisms which we cannot be able to see with our naked eyes, and to observe them, to see them, we require special optical device named as a microscope, are known as microorganisms or microbes. Who discovered them? Microbes was first discovered by Anton van Leeuwenhoek in 1674, when that scientist Anton van or A.V. Leeuwenhoek, A.V. stands for the short form of Anton van, discovered the first microorganisms, the first living cell, by observing the pond water in his self-made compound microscope. He was also the scientist who discovered the first microscope. Where do those microorganisms live? Microorganisms live almost everywhere on the planet Earth. They live in the most harsh conditions of the earth, like hot water springs, like marshes, like Antarctic region. In all those regions, the microorganisms exist in one or another form. Next thing is the 2 terms, what is microbiology and what is a microbiologist. Microbiology is the branch of science that deals with the study of microorganisms. That branch of science is known as microbiology. तो माइक्रोबायोलॉजी क्या है? ये एक ऐसी ब्रांच है साइंस की जिसमें एक साइंटिस्ट जिसका नाम माइक्रोबायोलॉजिस्ट होता है, वो माइक्रोऑर्गेनिजम्स की स्टडी करता है. माइक्रोऑर्गेनिज्म क्या है, कहाँ रहते हैं, उनका स्ट्रक्चर क्या है, क्या उनके हार्मफुल इफेक्ट्स हैं, क्या उनके यूजफुल इफेक्ट्स हैं. ऑल दोज आर इफेक्ट्स आर स्टडीड इन अ ब्रांच ऑफ साइंस, एंड दैट ब्रांच ऑफ साइंस इज नोन एज माइक्रोबायोलॉजी. एंड द साइंटिस्ट who does that study is known as microbiologist. So the scientist who does the microorganism study is known as microbiologist. Then what are the major groups of classification of microorganisms? There are in total 5 major groups of classifying the microorganisms. The first one is bacteria, singular is bacterium. Second is viruses, the singular is virus. Third, algae, the singular is alga. Fourth, fungi, the singular is fungus. And the 5th one is protozoa, the singular is protozoa. Then we have discussed all these 5 groups. First group we have discussed was bacteria. The singular is bacteria. Bacteria are the microorganisms which lives everywhere. They are the organisms which have a triple layer protective covering named as cyst. And due to that triple layer protective covering, they remain protected. They can survive in the most harsh conditions, in the most unfavorable conditions of the environment. Bacteria size is very small. They are the simplest and the smallest living organisms living on the planet Earth. The size of bacteria ranges from 0.2 to 1000 microns, and 1 micron is equals to 10 raised to the power minus 6 meter. The bacteria are of different types on the basis of their shape. For example, bacilli, that means rod-shaped bacteria. Example, Escherichia coli or E. coli. Next, cocci or coccus, that is rod or spherical, that is circular or spherical shape. The example is Streptococcus pneumoniae, the causative agent of pneumonia. Next, Spirulina or spiral-shaped bacteria. The example is Treponema pallidum, which causes sleeping sickness. Next, the 4th shape is Vibrio, that is comma-shaped bacteria, and the example of the Vibrio-shaped bacteria is Vibrio cholerae, which causes cholera. So these are the various forms of the bacteria on the basis of their shape. Second group, which we have discussed already, is viruses. The word virus is taken from a Latin word, and that Latin word virus means the poisonous liquid. Viruses are generally harmful for the human beings as well as for the living organisms. They cause multiple kind of diseases. Viruses are the connecting link between the living and the non-living organism. As Viruses possess the characters of both living as well as non-living things. Viruses can be crystallized, they can be solidified and crystallized like soil, which is a characteristic of non-living things. Viruses do not reproduce and grow on their own, which is a characteristic of non-living things. For the reproduction and for growth, viruses first indulge themselves into the body of host, into the cells of host, and then they burst the cell of the host and leave the cell and infect the other cells. And that's how viruses normally infect the whole body. Viruses cause certain kind of diseases. Viruses are of two types on the basis of their nuclear material, that is RNA viruses and DNA viruses. The example of various kind of viruses are corona virus, that is COVID-19, which causes corona disease, influenza, which is caused by influenza virus, AIDS, which is caused by HIV virus. So these are all example of viruses along with their diseases. The third class which we have done is algae. Algae are the most simplest plants found on the planet Earth. They have not a very complex body organization or cell organization as that of complex plants like trees of mango, banyan tree are. Algae do not possess the root, stems and leaves. They are flat, thallus-like, broad leaf-like structures only. They are not the leaves, they are the leaf-like structures. Algae usually found in the moist places. Algae differ in their size, shape and color. The algae are of different three kinds due to its pigment, its chlorophyll pigments present in their cells. Algae are autotrophic in nutrition. They can synthesize their own food by a process named as photosynthesis. The example of unicellular algae is Chlamydomonas, and the examples of multicellular algae are Spirogyra, Volvox and seaweeds, etc. What is unicellular and multicellular? The organism which is composed of one cell, single cell, is known as a unicellular organism, and the organism which is composed of multiple cells, many cells, is known as multicellular organism. So these are the two multicellular and unicellular terms meant for. The fourth and a very important class of microorganisms which we have discussed is fungi. The singular is fungus. Fungi are very well known for the saprotrophic mode of nutrition. They take their nutritions from dead and decomposing material. That is why they are also referred as the cleaners of the environment, cleaners of the earth, as they decompose the dead material and convert those poisonous material, convert those harmful material into the non-poisonous one, into the inorganic materials, which again mix with the soil and increases the soil fertility. Along with fungi, there are certain bacteria as well which performs the function of decomposition, which act as an environmental cleansing agent, which decomposes the material. So, if someone asks you a question, name the two microorganisms which decompose the dead and decaying matter, which helps in the cleaning of environment, the answer would be bacteria and fungi. Next one, fungi lacks chlorophyll. They are devoid of chlorophyll, that is, they do not contain chlorophyll. The green color pigment is not present in them. The example of unicellular fungi is yeast, and the example of multicellular fungi is usually used in our homes is mushroom. Next, bread mold or mucor, the green color pigmented fiber-like structure which deposited over the bread when we kept the bread in a house for quite a few days. That is actually a fungi, that is actually a fungus. So that is bread mold or mucor. Fungi are basically used in industries to produce wine and beer, that is the liquor industries, depends basically on fungi. The fifth and the last class which we have discussed is protozoa. Protozoa means the first animals. Protozoa have a very basic simple body design. They are unicellular eukaryotic organisms. Unicellular means single celled, and eukaryotes means their cell is composed of a nucleus. Protozoa basically found almost everywhere on the planet Earth, that is in the air, in the soil and in the water. Protozoa are specifically placed into a special kingdom named as Protista. I explained you in the previous video that total 5 kingdoms of classification are there for that whole living organism. The first one is Monera, the second one is Protista, the third one is Fungi, the fourth one is Plantae, and fifth one is Animalia. So the second kingdom, that is Protista, is having the protozoa. The example of protozoa is Amoeba, Paramecium, Euglena. There's one more, a very interesting thing about the Euglena is, Euglena shows mixotropic mode of nutrition. What is mixotropic mode of nutrition? Euglena contain chlorophyll. In the presence of sunlight, Euglena can prepare its own food. Whereas in the absence of sunlight, it shifts its nutrition towards parasitic mode, that it lives on the body of another living organism. So it shows 2 kinds of nutrition, autotropic, that it can make or it can synthesize its own food, and second one is heterotropic, that is in the absence of light, it shifts its nutrition towards the heterotropism. It takes its food from the other organisms. So that mode of nutrition is known as mixotropic mode of nutrition, and the example of mixotropic mode of nutrition possessing organism is Euglena. After the groups, we have discussed certain uses of the microorganisms. We are discussing that topic in between, the useful microorganisms. The first use which we have discussed in detail was formation of curd. The milk is set up into curd, and that setup of curd is done by the help of a bacteria named as lactobacillus. Milk contains a sugar named as lactose sugar, and that sugar is converted into an acid by the activity of lactobacillus. That acid is named as lactic acid. In the presence of that acid, a protein present in the milk named as casein coagulates, conjugates and form curd. So the coagulation of casein required acid and the formation of acid is done by the microorganism lactobacillus. From where that lactobacillus came, bache? We add a small amount of curd or buttermilk into the lukewarm milk so that curd can be set up easily. After that, we have discussed, bache, the formation of paneer and cheese is done by the help of streptococcus and lactobacillus respectively. For the formation of cheese, lactobacillus helps us, and formation of paneer, streptococcus plays a very important role. Today we will discuss in the next useful formation of any food item, and that is the tenderization of milk. Tenderization means softening of milk. There are certain kind of bacteria and yeast are used which softens the meat, and that softening of meat is known as tenderization of meat. The next use is fermentation. This fermentation we will discuss in detail in the next topic as well. But what is fermentation? The conversion of carbohydrates like glucose into alcohol, carbon dioxide is known as fermentation. The conversion of glucose into alcohol, that is ethanol, and carbon dioxide is known as fermentation. Louis Pasteur was the scientist who discovered fermentation. Next, food. Some algae are there or seaweeds are there which are used as a food items, and the example of those algae is chlorella, which is used as a seafood mainly in the Japan. Next, agar-agar. Agar-agar is a jelly-like substance which is obtained from algae, example Gelidium and Gracilaria. The two examples of the algae which provides us agar-agar, a jelly-like substance, are Gelidium and Gracilaria. That jelly-like substance is used in ice creams, in jellies and in squashes. We use them in puddings also. We use them for the softening purpose and for the taste purpose. So these are the usefulness of certain microorganisms in the food items. The second use of microorganisms is in commercial purpose. Microorganisms, especially yeast, is used extensively in the bakeries, in the production of liquors like wine and beer, as yeast performs a process named as fermentation. Yeast anaerobically converts the sugar into alcohol, that is ethanol and carbon dioxide, and this process is known as fermentation. The carbohydrates like glucose, by the activity of yeast is unicellular fungi, converted into ethanol. Ethanol is an example of alcohol along with carbon dioxide and energy. With that energy, the yeast survives, they reproduce and gain energy, and ethanol is used in the liquor industry, and this process is known as fermentation. The production of alcohol and carbon dioxide from the carbohydrates like glucose by the activity of yeast over them is known as fermentation. Fermentation was discovered by a scientist named as Louis Pasteur in 1857. He was the first scientist who discovered fermentation. So if someone asks you, name the scientist who discovered fermentation, the name of the scientist was Louis Pasteur, was the scientist who discovered the process named as fermentation in 1857. Rather than yeast, there are certain bacteria which we use in the production of tea, tobacco and coffee. So certain bacteria are used in the production of tea, tobacco and coffee. These bacteria are not used in the production of the plants of the tea. They are used to separate the tea from the tea leaves. Next, some bacteria are there which we use in the formation of certain acids, certain useful acids like lactic acid, citric acid and acetic acid, which we used as a vinegar. The next use which we are going to study is in the medicinal use, in the medicinal industry. In the medicinal industry, the first use which we have to study in the detail is the production of antibiotics. Before studying that, first we'll discuss what are antibiotics, how we get them, how we produce them initially. What are antibiotics? Antibiotics are the chemicals which are produced by certain microorganisms, especially bacteria and fungi, which are used to kill the pathogens. Now the question arises again, what are pathogens? The disease-causing microorganisms are known as pathogens. Any microorganism which causes a certain kind of disease is known as a pathogen. ऐसा कोई भी माइक्रो ऑर्गेनिज्म बच्चे जो हमें डिजीज कॉज कर सके उसे हम क्या बोलते हैं? एक पैथोजन बोलते हैं। और उन पैथोजन को मारने के लिए हम कुछ केमिकल ऑब्टेन करते हैं, किससे? बैक्टीरिया और फंजाई से। उस केमिकल को हम क्या कहते हैं? एंटीबायोटिक कहते हैं। सो द मेडिसिन्स प्रोड्यूस्ड बाय द लिविंग ऑर्गेनिजम्स, एग्जांपल और सच एज बैक्टीरिया एंड फंजाई, व्हिच आर यूज्ड टू किल the pathogens, that is disease causing microorganisms, is called antibiotic. There are various kind of antibiotics are known to date. More than 150 antibiotics we have discovered till date, out of which few examples are streptomycin, aueromycetin, tetracyclines and penicillin. Penicillin was the first antibiotic which was discovered by Sir Alexander Fleming. Accident. How we discovered it? Actually, he was growing a culture. He was growing a tissue of a bacteria named as Staphylococcus. Alexander Fleming क्या कर रहे थे बच्चे? बैक्टीरिया के कल्चर को ग्रो कर रहे थे. Now the question rises, what is culture? बैक्टीरिया की कॉलोनी को, बैक्टीरिया के नंबर को बढ़ा रहे थे. Accidentally, a spore of a fungi named as Penicillium notatum fell over in that culture. एक्सीडेंटली क्या हुआ? अचानक से क्या हुआ? उनसे या एक स्पोर, किसका स्पोर? एक फंगस का स्पोर, उस फंगस का नाम है पेनिसिलियम नोटेटम, गलती से उसके अंदर गिर गया। एंड ही ऑब्जर्व दैट नॉट दैट फंगल स्पोर ग्रो, सिर्फ वो फंगस ग्रो नहीं हुआ, उस फंगस ने क्या किया? दैट फंगस ऑल्सो kills that bacterial culture. उसने उस Staphylococcus bacteria को भी मार दिया. Then he came to a conclusion that that fungal spore must contain certain chemical which kills that bacteria. तो उन्होंने क्या conclusion निकाला कि इसके पास एक ऐसा chemical है जो उस बैक्टीरिया को किल कर देता है. And he discovered the first antibiotic named as penicillin. So the penicillin was the first antibiotic which was discovered by Sir Alexander Fleming from a fungi Penicillium notatum. Now we'll discuss certain precautions which are to be taken care of while taking the antibiotics. You can't just take the antibiotics anyway. You must have to use the antibiotics only by the advice of a qualified doctor or a specialist doctor. Until a doctor will give you advice or ask you to do so, until then don't eat the antibiotics. Because sometimes antibiotics, when taken unnecessarily, may kills the beneficial bacteria which is used to live in our intestine. As antibiotics used to kill the microorganisms, and certain microorganisms are present in our body as well, which are human-friendly microorganisms. So unnecessarily taken antibiotics may kill those bacteria. So the first is they must be taken by the advice of a doctor or under the surveillance of a doctor. Next, do not take antibiotic in excess. Why? Because the wrong doses or excess doses may put adverse effects on our health. Certain antibiotics are very strong and that may harm our liver. So, in excess dose of antibiotics must be avoided. Next is entire course must be taken. If, for example, we started the use of antibiotics and those antibiotics are for 10 days. That those are suggested to us for the 10 continuous days, and we stop those antibiotics in ingestion after 5 days. That means we are not completing our course. In that case, there are chances that that bacteria, that pathogen may grow again. So we must have to complete the entire course of antibiotics which is suggested to us, which is advisable to us by a very qualified doctor. The next is unnecessary use, as I explained you. Unnecessary use of those antibiotics may kill the beneficial bacteria of our body which used to live in our intestine. Next one, check. In case of flu, in case of common cold, do not use antibiotics. Why? Because the flu and common cold are viral diseases and antibiotics are not effective against the viral diseases. Why is it so? Because the viruses do not have their own metabolism. So that is why antibiotic drugs are not effective against viral diseases.